And so as I had mentioned, about 5% of our global melatonin is produced by the pineal gland, and it's related to the circadian cycle. 95% of the melatonin, though, is produced locally inside all of our cells that contain mitochondria. And the only cells that don't contain mitochondria are the RBCs, essentially. They're not producing melatonin, but every other cell is producing melatonin. And that cellular intramitochondrial melatonin does not contribute to any significant quantity of melatonin that we get in the circulation. It's all consumed and used locally. And so what does it do? I mean, why do we have an entire system in the mitochondria that generate this melatonin locally? And why is it so conserved that it can't get out of the cell? And what it does is it's a potent reactive oxidase scavenger. It pulls the free radicals that are generated during oxidative phosphorylation, and it quenches them. Because if it doesn't quench them, you activate cytochrome C and you trigger an apoptosis, and you can cause immediate dysregulation. The better your antioxidant status with regards to melatonin as a scavenger of free radicals, the less damage that you're going to create in the mitochondria. And that issue associated with the proton gradient that exists between the inner and the outer shell of the mitochondria, the inner and outer membrane of the mitochondria. So the melatonin accumulates in the mitochondria, and there's a whole host of methods for the production, but we've kind of touched on it. And the critical factor in the production of this is the presence of acetyl-CoA in sufficient quantity to trigger those critical enzymes that we talked about in melatonin synthesis. AANAT is the primary one. And it needs a cofactor, acetyl-CoA. So if you think about this for a second, and is that in the pathway of converting glucose to pyruvate, which happens in the cytosol, and then the pyruvate is transferred into the mitochondria, and it is manufactured into acetyl-CoA. That acetyl-CoA is the critical factor to convert serotonin into melatonin inside the mitochondria. Without sufficiency of acetyl-CoA and without sufficiency of the pre-existing tryptophan to serotonin, you're not going to get enough melatonin. And if you don't get enough melatonin, you can't scavenge the free radicals. So it posits an interesting question, because if you look at cancer cells specifically, they don't utilize the mitochondria as much. And they don't produce as much acetyl-CoA. They typically will consume the glucose in the cytosol, and they don't use the derivative properties of the oxidative phosphorylation system. And their melatonin level is dramatically reduced. And they have to they don't get to consume as much oxygen because they can't. And so when you do a PET scan, positron emission tomography, and one of the markers that we use for that is a labeled glucose. That's how you identify which cells are hyperactive with glucose uptake. It's because they don't get to produce as much ATP, and they're predominantly just shunting glucose to pyruvate in the cytosol and not getting as much efficiency as regular cells are that can actually get through the TCA cycle. So it's just something, and if you look at it that way, you also realize then that it's that those group of cells that are cancer cells are also not producing as much acetyl-CoA. And if they're not producing as much acetyl-CoA, they don't have the buffering system for a lot of the stuff. There's also some significant information about MS and the inflammatory demyelination that's associated with the mitochondrial enzyme instability, specifically related to melatonin production. It'll be in the notes on this particular slide. There's some lots and lots and lots of research that's done on that, and that may be a major factor for that. So near infrared and infrared wavelengths are absorbed by cytochrome C oxidase, and it's in the electron, it's in the factors 1, 2, 3, 4, and then into the ATP synthetase. That cytochrome C oxidase, where there's an interaction between the infrared light and the electron chain. These wavelengths improve the mitochondrial membrane potential. And when you activate that, that's what causes the melatonin production inside the cell. Thank you.